Well, everyone, hello, and you are back for another exciting lecture in biology. And today we're talking about something called classic genetics, which is also called the genetics of inheritance. If I can turn on my handy dandy wireless drawing tablet, brought to you by XP Pen. All right, so, anyways, I'm not sponsored by anyone, unfortunately. If I was, though, it probably would be a drawing tablet manufacturer. So, anyways, I need to change the way that I teach my bio classes because I'm like teaching the same exact thing now with all the bio classes that I teach. That's a lot of them. I find myself telling the same corny jokes at the same parts of the same lectures, which I don't know if that's a compliment of how well I've been planning my teaching or if it's a detraction because now I feel like I'm not telling my jokes with the same gusto as I'm accustomed to. So I'll let you be the judge of that. Maybe I'll throw in a joke this time that's not planned in advance. We used to make fun of my math professor at Moore Park, Mr. Walter. I don't think he still teaches there, but um, we used to joke, my friend and I who took the class, that he planned his jokes during lecture, and we would talk about, yeah, it's like as soon as we get to, I don't know, binomial equations, it's time for a story, and we were laughing, ha ha, figured out the teacher's trick. And I'm the teacher that plans his jokes at times in lecture. Be careful, you people that are considering teaching. You criticize and judge because of my joke insertions. You might be there one day too, planning your jokes. All right, so unfortunately, this is not Seinfeld Comedy Hour. If you don't know who Seinfeld is, I suggest you Google it. But anyways, today we're talking about classic genetics, which is also known as the genetics of inheritance. And if you remember, in meiosis, is one diploid cell split into two haploid cells. And just to give you an example, we'll say that this is parent 1 and this is parent 2. Now, we know that each parent has two chromosomes of each type. Now, when I say type, chromosome 1, chromosome 2, chromosome 3, so on and so forth. Chromosome 1 has certain genes, chromosome 2 has certain genes, so on and so forth. However, each parent has two of the same type of allele. You recall they're called homologous chromosomes. So to give you an example of how inheritance works, we're going to go ahead and say that parent 1 and parent 2 both have one copy of chromosome 1, and each one has a different variant of the gene for eye color. As we'll talk about shortly, an allele is a variation of a gene. Now, I like to have some fun with this, so don't be surprised to see some kind of wonky examples here. Let's say parent 1 has a chromosome with an allele for blue eyes. And let's say, so we'll say blue eyes and one for black eyes. Wow, okay, I just realized they both start with B. Can't have that. Purple eyes. And we're going to go ahead and say that parent 2 has the same exact alleles, purple and blue. And if you remember, is after meiosis, when we have four haploid cells, there's only one copy of each chromosome in these haploid cells, like so. So drawing this. All right, and we'll go ahead and just say that in this gamete, because recall that when haploid cells are made, once again, there's only one of each chromosome. So we have blue, and we have purple. So what this means is that when these two parents decide that they are going to have offspring, the type of chromosome, or how should I put this? The haploid cells that become fertilized are random, meaning that you could have the possible haploid cells combine and form an offspring. So these are the options for inheritance. Perhaps this sperm containing the purple eye allele will fertilize this egg that has the blue eye allele. Therefore, the combination of alleles will be B and P, so blue and purple. Or maybe this haploid cell will fertilize this haploid cell and we'll have purple, purple. Or maybe this haploid cell will fertilize this haploid cell and we'll have BB. So there's actually 3 different possible combinations for the type of alleles that this offspring inherits. Now you also can have this combination here, which is this B allele and this P allele. And you're probably saying, okay, well, why are you doing this? We already have all the combinations. Well, that's because there's 4 possible ways that these alleles can be combined in the new offspring. But if we look here, out of these 4, these 2 are the most common, meaning that if this person has X number of kids, let's say 20 kids, we'd expect actually that 50% would inherit the 1B and 1P allele combination. 25% chance would inherit 2 purple eye alleles, and 25% chance will inherit 2 blue eye alleles. And this is exactly what we are doing here, is we're going to talk about the probability of inheritance patterns in offspring. That's what classic genetics is, really, is it is the probability of an offspring inheriting a certain combination of alleles. That's basically it. Now, of course, it's much more complicated than that. If you decide that you really like this genetic stuff, one of the most fascinating college courses that I took was genetics. At CSUN, it was a 300 level class, but it was really fascinating, and the professor I had was just awesome. But yeah, we're just doing a basic introduction. If any of you took AP Bio in high school, this is probably going to be simpler than what you learned. But yeah, it will give you a nice broad overview. So first, we need to talk about some essential terminology. And the first thing, I don't even want to write this down, but chromosomes. If you remember, our DNA is split into 23 unique pieces. However, we have 2 of each chromosome, so that's 46 unique pieces. So each chromosome contains different genes. In fact, what am I doing? Stop being lazy, Thomas. All right, so chromosomes. If you recall, these are segments of DNA containing unique genes. So with us humans, we have 23 unique chromosomes. I think last lecture I talked about, or lecture before, how I talked about how chromosomes are like pairs of shoes. Is you can have 23 unique pairs of shoes, but you got 2 each, so that's 46 shoes total. So we have 23 unique chromosomes, but we have 2 of each, which is going to give us 46 total. All right, so I think I also showed you before that, let's say that we have this chromosome here, and this is extremely simplified. Chromosomes have dozens and dozens of genes on each chromosome or more. But I'm going to say that this one controls eye color and hair color. So I'll just say eye and hair color. So we'll say that this is chromosome 1 for an individual. And we'll go ahead and say that the eye color is going to be pink. And the eye color here is going to be blue. We'll say that for here, we have, I don't know, orange. And gosh, I kind of backed myself into a corner here. Orange and we'll say red, whatever. So one individual has inherited these 2 chromosomes, each encoding a slightly different gene called an allele. So if we're going to write here a gene, well, actually, let me write this down here. So a gene is a segment of a chromosome encoding a single protein or trait. Encoding a single trait. So we see here that the gene here, now the gene is not the allele. The gene is just any part of a chromosome that codes for a specific characteristic or trait. So an example of a gene would be, for example, eye color and hair color. However, an allele is a variation of a gene. So an allele is a gene variant. So you could say that a gene is analogous to a car. And an allele is analogous to a type of car, like Toyota, Subaru, Suzuki. Do they still make Suzukis? I don't think so. I'll have to double check that. So yeah, an allele is a gene variant. So for example, when we talked about, oh no, disaster has struck. The XP pen has run out of charge. Don't worry. I can now switch from wireless to wired mode. Let me just figure out how. Yeah, okay. Yeah, there was a funny commercial back in the 80s with a salesman named Suzuki Joe. And he would be on commercials just making outlandish claims like, oh, a Suzuki can out-sprint a Ferrari in 0 to 60. And then there would flash on the screen, he's lying. And he'd say something like, I don't know, Suzuki has more comfortable seats than the Rolls Royce. And then he's lying, and it should be, ha ha ha, he's lying about Suzuki's because it really can't be the Ferrari. Yeah. Okay. Anyways, an allele is a gene variant. So, for example, pink eyes and blue eyes. So we are all very complex organisms. Because of that, we all have 2 alleles for each trait, 2 alleles for each gene. Try not to confuse my terminology too much. Now, there are some genes that really do not have alleles with them. And that's because if they mutated, if they changed over time, yes, in case you're curious, is mutations, random changes in DNA. They are the ones responsible for different alleles, just slight mutations. But there's some that if they change even a tiny bit, they're no longer functional, and that they make something that's really important, like hemoglobin, for example. If that changes too much, it can't do its job of carrying oxygen, and the organism cannot survive. But thankfully, there's a backup system for that, which we'll talk about in a bit. A trait is anything that we call a observable characteristic. A trait is very similar to a phenotype, which we'll talk about in a moment. Observable characteristic. For example, eye color and hair color. How is this different from a gene? Well, a gene just contains the information for. So if we're going to keep with our car example, a gene might contain the instructions for making a Honda Civic. A trait, though, is actually the already constructed Honda Civic. If you don't like that one, think of a recipe. Like a recipe for chocolate chip cookies would be a gene. The actual chocolate chip cookies would be a trait. Now, when we talk about classic genetics, get used to hearing two terms a lot. One is going to be genotype, and the other is going to be phenotype. And a genotype is the combination of inherited alleles. And so, for example, the genotype for this individual would be pink-blue for eye color. So, for example, it would be pink and blue. That would be the genotype, because they've inherited one allele for pink eyes and one allele for blue eyes. Now, the next is phenotype. Phenotype. And a phenotype is the visible outcome of the allele combinations. Now, depending on the method of inheritance, the phenotype is going to be different. Now, in some cases of inheritance, the pink and blue eye alleles will actually combine to form a blend. Or maybe they'll both be visible on the individual, like pink and blue polka dotted eyes. Or maybe one allele will completely overshadow the other. So the individual will only have pink or blue eyes. So you're seeing that inheritance is incredibly complicated. The genotype is easy. They have one allele for pink eyes and one allele for blue eyes. The phenotype, though, remains to be seen because it all depends on how the alleles interact. Now, there's some people that have, like, for example, mixed hair colors. They'll have what's called dirty blondes. And that's because their two alleles for hair color interact to form that phenotype. However, some people have one gene that's functional, one gene that isn't. Because of that, the gene that doesn't work is completely hidden. We'll talk about that too. It's actually pretty interesting. Okay, now I couldn't fit everything on this page, unfortunately, but we have two other terms here. And this is going to be continued terminology. We have a term called homozygous. Very important here, homozygous. What this means is that two of the same alleles are inherited. Same alleles are inherited. All right. We also have a term for this. It's also called true breeding. Now, there are two types of homozygous inheritance, and one is called dominant. One is called homozygous dominant. This means that two functional alleles are inherited. This actually goes along the lines with, well, actually, no, we're going to hold off on this because it's going to be confusing, because we're going to talk about this again when we get to dominant recessive inheritance. All right, the other type is called heterozygous, meaning that two different alleles are inherited. Two different alleles are inherited. Alleles are inherited. Now, what this means is that if an individual inherits two different alleles, the phenotype may is the key word, may be a new see, I'm trying to think of a way to put this where it will not be confusing. I'll just put down the interaction between the two different alleles. The two different alleles can create a third phenotype. You'll see what I mean later, but it's important to keep in mind that the heterozygote is usually a wild card, meaning that if there's two alleles, for example, in the combination, in the population, sorry, the heterozygote could be a brand new phenotype. Wow, that did not work out well. Oh, no, not work out well. I was going to try and move this to the left, but that's not going to happen. Yeah, it could either be a new phenotype or it could be the same as the dominant phenotype. So I'll just go ahead and put this down now. Is in, because this is, lots of students get confused on this, so I'm just going to go ahead and throw it out now. With dominant recessive inheritance, dominant and recessive inheritance, heterozygotes has the dominant phenotype. I'm sure that you remember some of this from your high school bio class, but we'll be reviewing it, don't worry. However, in incomplete and codominance, heterozygote is a new phenotype. Heterozygote is a new phenotype. Definitely keep that in mind because lots of students get confused with the heterozygote. The homozygote's easy because if you have an allele for purple eyes and purple eyes, if that's the genotype, you're going to have purple eyes. Easy peasy. But if you're the heterozygote, then it gets more complicated because if you have an allele for purple eyes and an allele for blue eyes, there's no guarantee just by looking at the genotype that you'll be able to predict the phenotype. So the first type of inheritance we're going to talk about is called dominant and recessive inheritance. Now, you probably remember Mendel and his pea plants. And I think that the story of Mendel is fascinating. Is you have a monk who more or less was just cross-pollinating pea plants, and he basically found out the idea of genes and alleles and inheritance patterns. So that was amazing. I don't spend too much time on the historical aspects of it, but he more or less discovered dominant recessive inheritance, which is amazing. But anyways, dominant and recessive inheritance is inheritance patterns where a functional and mutated non-functional alleles are present in a population. An example of this is, or this is usually whenever you discuss inherited genetic disorders, it's almost always an example of dominant and recessive inheritance, because certain alleles exist in certain populations that have been mutated and do not work. They're non-functional. To give you an example of this, we have a couple of disorders here. Let me go ahead and show you. So this is normally if you want real examples of dominant recessive inheritance, is usually your go-to is going to be inherited genetic disorders. And so I'm going to go ahead now. Let's look at. Yes, when this video was recorded, it's Valentine's Day. You can tell that my Valentine's Day is just riveting. Went to Target, bought some cleaning products so I can make sure that at the high school I teach at, it's COVID-free. Don't trust students. And now I'm giving a lecture on classic genetics. All right, so let's go ahead and you can tell also the things I've been Googling have just been also riveting. All right, so we're going to type in cystic fibrosis. And this is in a disorder where an organism inherits two alleles that are mutated that code for a type of protein called the cystic fibrosis transporter protein. And if this receptor is mutated, which if you just want a sort of a brief overview, if this receptor is mutated, as in the individual has inherited two alleles that do not properly code for the CFTR protein, then this individual will not be able to make watery mucus. This protein must be functional for watery mucus to be made. Because of that, people that have inherited cystic fibrosis, because they have two non-functional alleles for the CFTR protein, is they have very thick mucus that is very hard to bring out of the airways, and it ends up actually sinking down into the lungs, causing lots of infections. So that's an example of an inherited recessive disorder. So you can see here that the thick mucus is dripping down into the lungs, restricting airflow and causing infections. Another example is going to be something called albinism. I cannot spell albinism, okay? You see here that these individuals, the gene to code for the presence of skin and hair pigment is mutated. If two parents pass down a mutated allele, which we call recessive, for skin and hair color to their offspring, then their offspring will have albinoism. And if you look here, these individuals do not have functional gene that codes for hair color, skin color, and eye color. Why is this? Well, because their parents may look normal on the outside. They may be phenotypically normal, but each of them was carrying a recessive allele in their genome, meaning that they have one allele for functional pigment color and one allele that's mutated and has non-functional coding for pigment color. So these individuals inherited the two non-functional alleles called recessive. So I know that's kind of confusing, but I'll clear it up, don't worry. All right, so what this means is that dominant and recessive allele, or inheritance, is one allele is functional and the other allele is non-functional. Meaning that the allele that we call dominant, which we assign a capital letter to. So dominant is a functional gene that creates a functional protein. Functional protein. If we say that an allele is recessive, it means that it is a mutated gene that does not create a functional allele. All right, now I just showed you some recessive disorders, and albinoism is not life-threatening, even though they have to be careful about going outside without proper skin protection because skin pigment actually protects against UV rays. But cystic fibrosis is life-threatening without treatment. So people often ask, why is it that recessive disorders like cystic fibrosis can persist in the population? And that's because normal individuals, so one thing to keep in mind is phenotypically, normal individuals can carry, very important term here, carry a recessive allele. Because with dominant and recessive inheritance, as long as an individual just has one dominant allele, phenotypically they'll be normal or mostly normal. However, they can carry the recessive allele, meaning if they happen to mate with someone that is also carrying the recessive allele, they can pass it down to their offspring. Now, whenever we refer, oh yeah, I promise you we talk about dominant recessive inheritance, the term homozygote and heterozygote. So in dominant and recessive inheritance, the homozygote that we call true breeding, there's two types, because remember homozygote means two of the same allele. So one type is we call dominant. And dominant means that two functional alleles are inherited. Now, this also means that they have the dominant phenotype, obviously, because they have two dominant alleles. Now, also, they cannot pass down any disorder to their offspring because they don't have a disordered allele. So, offspring will always have one functional allele. Will always have a functional allele. I know this for some of you, if this seems obvious, that's awesome because that means that you understand this genetic stuff pretty well. If you're learning a bunch of cool stuff for the first time, then awesome. So yeah, when I teach, I do a lot of repetition because I know that I tend to space out sometimes. Chances are you are listening to this or watching this lecture. Maybe you're engrossed in every word, but some of you also might, I don't know, be texting your friend about something or TV's on in the background, something like that. So figure if I repeat something a couple times, then either it'll be properly reinforced, you'll be catching it for the first time. So recessive, if you are what we call homozygous recessive, two dysfunctional alleles are inherited. Now, this has the recessive phenotype, meaning that the individual is missing something that they need in order to properly function. And also, offspring will always have a recessive allele. A common question I get is, how is it that that individuals that have two recessive alleles for a trait, how is it that they're able to reproduce? Because shouldn't they be relatively non or in too ill of health to reproduce? Well, sometimes, for example, with albinoism, is they can survive just fine with some modifications. But there's other recessive disorders that actually don't appear until later on in life. So somebody could be phenotypically normal until their 30s, 40s, or 50s even, and then there's the onset of the condition. All right, now next is going to be the heterozygote. And if you remember, the heterozygote has two different alleles. And in dominant and recessive inheritance, the heterozygote always, very important, always has the dominant phenotype. But there's also a 50% chance of passing the recessive allele to their offspring. Think of it like this. So let's say that you have two pencils on you. They're mechanical pencils, so they look the same. One has lead, one doesn't, and you don't know that. You assume that both pencils have lead. That is how a carrier lives their life. Somebody that carries the recessive allele. Um, they think that everything's fine. But let's say that a friend would be to ask for a pencil and you were to give them a random pencil and it would be the one that doesn't work. Your friend was well intentioned, but one of the pencils he or she gave you was non-functional. And now you're slightly resentful because maybe you needed that pencil for your math test, but a heterozygote is always a carrier for recessive allele. And usually they don't know that. Carrier for recessive allele. Usually they don't know that. In fact, they really only find out when if they have offspring with somebody that carries the same recessive allele, or the individual has the disorder themselves. Now I'm going to give you some sample genetics problems. We do things simple in this class. It's called a monohybrid cross. And a monohybrid cross is the probability of parents passing down a certain combination of alleles. Usually in genetics problems, you get things like flower color, pea shape, corn shape, whatever. So I tend to mix it up. Not all the problems are factual, but the math, quote unquote, is the same. All right, so let's go ahead and write out a problem here, and then I can sort of explain to you how to solve these. Okay, so we're going to go ahead and say that a florist has flowers, fitting for Valentine's Day, flowers with red petals and flowers with white petals. She cross-pollinates, meaning she takes the pollen from one flower and gives it to the other flower. She cross-pollinates these two flowering plants. Okay, one plant is heterozygous for purple color. The other plant is homozygous for white color. What is the phenotypic probabilities? Okay, so this florist has flowers with, wait, what the? Okay, I meant, oh gosh. Well, yeah, hold on a second. Gosh darn it. Well, let's go ahead and say easy fix. All right, now the first thing that we do is we create a legend. And you're probably saying, well, what's a legend? A legend is you are going to assign a capital and lowercase letter for each possible phenotype. So in this case, our trait is going to be flower color. Now, when I see a capital letter, I mean, for example, big A is going to be referring to the dominant allele, and small a is going to be referring to the recessive allele. That is to remind us that one possibility is dominant, the other is going to be recessive. Now, you might be saying, well, wait a second, professor, you didn't tell us which one is dominant and which one is recessive. And the answer is yes, I did. Because I said that this plant is heterozygous for purple color. And also recall that the heterozygote always has the dominant phenotype, the dominant allele. So in this case, we know that purple is the dominant allele, the functional one, because the heterozygote always has the dominant phenotype. So in this case, purple is going to be our dominant allele, and white is going to be our recessive allele. Now, the next thing we do is we create, well, actually, create's not the right word, is we write down the parental genotypes. So P1 equals, so we know that plant 1 is heterozygous for purple color. So we're going to use big A and small a, because recall that each organism has 2 alleles for the same trait. And we put an X to mean cross with this flower that is white. So we've already established that white is our recessive allele, and we also talked about that in order for the recessive phenotype to be displayed, 2 recessive alleles must be inherited. That's the only way that the recessive phenotype can be displayed, if the organism has inherited 2 recessive alleles. And now you are going to create a Punnett square to find out all the possibilities. Actually, you are not. I haven't done a Punnett square since Ronald Reagan was in office back in the 80s. All right, I'm exaggerating a bit, but I'm not a fan of the Punnett square, not because it isn't good, but because it isn't fast and it's not as accurate. Once you get to the point where you're doing tri-hybrid crosses, which has 64 possibilities, you don't mess with the Punnett square anymore, unless you really like filling in 64 boxes. So I use something called the foiling method, which is similar to what you learned in math. Is you are pretty much just going to cross multiply these. Well, not multiply, but if you remember FOIL, you go first. So the first allele here and the first allele for parent 2. So one possible combination is big A, small a. Now, in a monohybrid cross, there's a 25% chance or 1/4 chance of getting each of these outcomes, because in monohybrid crosses, there's always 4 possibilities. So there's a 25% chance that the organism can be heterozygous. Now we do outer. So first, then we have outer. So another possibility is 1/4 heterozygous. And if you're astute, you probably have picked up that these are exactly the same. They are. We'll deal with that in a moment. Then we have inner. So that's about 25% chance of being homozygous recessive. And then we have last, which would be small a, 1/4 chance, small a. So now we look and say, are any of our 4 outcomes the same? And actually, yeah, 2 of them are the same. So since these are the same, we have to combine the probabilities. 1/4 plus 1/4 is 1/2. Or if you want to deal in percentages, 25% plus 25% is 50%, but I tend to deal in fractions. So there's a 25% chance of an offspring inheriting being heterozygous. There's a 25% chance, because we have 2 of the same outcomes here. So 25% chance of the organism being homozygous recessive. So 1/2 small a. So the 2 outcomes are either going to be, because these will give us 2 different phenotypes. there's a 50% chance of an individual flower being purple and a 50% chance of it being white. What this means is that if you were to cross-pollinate flowers and have 1,000 offspring flowers, you'd expect 500 should be purple, 500 should be white. Okay. Now, let's go ahead and change this problem slightly. Let's go ahead and say now, oh, I should have said 3 is going to be foil, in case you're curious. So now we are going to say we're going to change this slightly. Cross 2 heterozygous purple flowers. Okay, easy, easy. And we'll say that, let me see if I can select this text box. Determine the phenotypic probabilities. Okay, no problem, but this is just a little bit trickier. So the parental cross is going to be like this, big A, small a, big A, small a. And if we're going to foil, it's going to look like this. So first, outer, inner, and last. Now we see that there are 2 possibilities for an offspring to be heterozygous. So this will turn out to be 1/2 possibility of an offspring being heterozygous, and 1/4 being homozygous dominant, and 1/4 being homozygous recessive. Now, even though there's 3 possible genotypes, there's only 2 possible phenotypes, right? Purple or white. Now, we see here, if you recall, that with dominant and recessive inheritance, as long as one dominant allele is inherited, then that offspring will express the dominant phenotype. So we have here 2 genomic combinations that have at least one dominant allele. So in this case, since we're concerned with the phenotypic possibilities, 1/4 plus 1/2 equals 3/4 of a chance for a flower being purple and 1/4 chance of flower being white. That's because, once again, with dominant and recessive inheritance, both the dominant homozygote and the heterozygote each have one dominant allele, at least one dominant allele, meaning they'll express the dominant phenotype. All right, but that is going to be a little bit more complicated than what we wanted to. Well, actually, this is simpler than what we want. Well, sorry, this is a simpler form of inheritance. So now we're going to have another type. So now we're going to have complex inheritance patterns, and we're going to start with incomplete and codominance. Now, incomplete dominance is the heterozygote is a third phenotype. Now, with incomplete dominance, the heterozygote has a blended phenotype, like mixing 2 colors of paint. And also, it's important to keep in mind both alleles are functional. Because they're both functional, the phenotype that they create in this case is going to be a blend. So to give us an example, flowers can either be, what color should I use? Blue or yellow. Blue, yellow, or green. So your main hint that this is an incomplete dominant problem is that there's 3 phenotypes, and green is a blend between blue and yellow. If a green and yellow flower are crossed, what will be the phenotypic outcomes? All right, well, let's go find out. Remember, before you dive into any genetics problem, always do a legend. No, draw. Good. So always do a legend. And big A is going to be, we'll say that big A is going to be blue. Now there is no small a. Now we have a different capital letter. Big B is going to be yellow. Okay, I probably should have made blue big B, but whatever. So you're probably saying, well, wait a second. What's the combination for green? Where's the allele for green? There isn't one, because since green is a blend between blue and yellow, actually, if you are going to come up with a legend for green, it would be AB equals green. So as I said, your main hint that you are given an incomplete dominant problem is when the third phenotype is a blend. So if we cross a green flower, which will be big A, big B, times a yellow flower, big B, and we do FOIL, we'd have first 25% chance green, outer, 25% chance green, inner, 25% chance yellow, and last, 25% chance, oops, got ahead of myself, 25% chance yellow. And of course, we need to combine a like combinations. So 50% chance yellow and 50% chance green. So once again, these problems are going to be similar, but with incomplete and codominance, there is no recessive allele, no lowercase letter. Both alleles create a functional phenotype. However, the heterozygote is going to be a blend. Now, the other type is going to be called codominance. And with codominance, two alleles interact and create a phenotype where both alleles how should I put this clearly? Where both individual phenotypes are visible. Once again, also, the heterozygote is a third phenotype, and each allele creates its own phenotype. Okay. So what does this mean? Well, let's go ahead and do a problem. Flowers can either be blue, yellow, or blue with yellow spots. If a flower that is blue, if two flowers with blue and yellow spots are crossed, what will be the possible outcomes? Now, in this case, the problem, as you can see, the problem is basically the same, where we have blue and yellow flowers. Those are the two alleles in the population. However, you know that this is an example of codominance because the third phenotype, you can see both the blue and yellow alleles present. So whenever you can see each individual allele, that is going to be codominance. Now, the legend is actually going to be the same. I'm not going to use blue again. So we'll say that big A, once again, is going to be blue. Big B is going to be yellow. And if we are going to do a cross here with two flowers that are blue with yellow spots, if we're going to FOIL first, outer, inner, and last. Combine alike genotypes, we end up with 1/4 of homozygous for blue. We have 1/2, which is going to be heterozygous with blue and yellow spots. Then we have a 25% chance or 1/4 possibility for homozygous for yellow. So what this means is 25% chance blue, 25% chance blue, yellow spots, and a 25% chance of being yellow. So you're seeing here that the genotypes are the same. The alleles are both functional. So with incomplete and codominance, the alleles are both functional. The difference is how the genes interact in the heterozygotes. With codominance, there's not a blended phenotype made. Instead, each of the alleles are separately present. So another example would be if you were to have a cow with a black coat crossed with a cow with a white coat. If it's in complete dominance, then the cow will be completely gray. If it's co-dominance, the cow will be white with black splotches. So that's an example of incomplete versus co-dominance. The PowerPoint has blood type as an example. It's very interesting. It's fascinating. If you take physiology, I'd be happy to. Well, that's one thing that we'll talk about. But in this class, it's optional in the PowerPoint if you want to check it out. Now, sometimes inheritance can be pretty complex. So I'm going to jump to the PowerPoint now because it has much better imagery. Pardon. All right, let me see if I can easily find this because my desktop is an absolute disaster, to put it nicely. Uh, okay, so welcome to my PowerPoints. I'm actually very proud of my PowerPoints. I used to teach exclusively by PowerPoint, and then I realized that when I was teaching physiology, I actually switched to doing just board diagrams, and my test averages jumped up like 10%. And I feel that it's because when the teacher writes out things, the students also write out things, and we learn best by doing. So I don't exactly draw here because I have a passion for for expressing my art to the world. I do so because it forces you to take notes. Well, that's my plan. It highly encourages you to take notes, and then as a result, you learn better. All right, so the first type of complex inheritance pattern is called pletrophy. In this case here, one gene actually controls multiple phenotypes. And an example here is seen in what is called a white tiger. And I say that in extreme quotation marks because a white tiger is actually a Bengal tiger with a mutated SLC45A2 gene. If this gene is mutated and non-functional, it actually causes the Bengal tiger to instead have a white coat, and it also causes them to be cross-eyed because this gene controls hair coloration and eye-brain wiring. So pletrophy, one gene controls multiple phenotypes. Now, the next type of inheritance is called polygenic inheritance. Poly for many, genic for genes, many genes. In this case, one characteristic is controlled by multiple genes. Because of this, the phenotype is generally seen on a bell curve, meaning that depending on the combination of genes and genes inherited, you can look more similar to one parent, more similar to the other parent, or somewhere in the middle. An example is if two parents were to create offspring, one that's very light complected and the other that's very dark complected. Since skin color is comprised of three genes, the way that these genes interact determines the phenotype for the child. So we see here, this is that famous tri-hybrid cross I was telling you about with 64 possibilities. We see that there's a very small chance, a 1 in 64 chance, that a child can have the same pigmentation as the mom or the same pigmentation as the dad. There's also lesser possibilities that the child can have a skin pigment that's more similar to one parent or the other parent. More than likely, the child's going to have a blended phenotype where they kind of look in the middle between each parent. So if you have siblings or you know a family and one child is like, for example, light complected, the other is darker complected, that's an example of polygenic inheritance. Next is going to be a pedigree chart. And really the only thing that I want you to know with pedigree charts is that is how to identify them. It's so one thing to note here with the pedigree charts is that, let me show you a bigger one. So what it is that it shows you the um, the presence of a certain genetic disorder in a family lineage. How it's shown is that if a parent has um, the disordered phenotype, then that box is going to be colored in. If the box does not have the disordered phenotype, then that box will not be colored in. So we see here that there's a circle and a square that's connected by a horizontal line. Now, circles refer to females and squares refer to males. So we see here that the parent is going to have whichever disorder is in question. The uh, the 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 grandmother here, because this is divided in generations. The grandmother has the disordered phenotype. The grandfather does not. They have three offspring here. They have a son, a daughter, and a son. And neither child has the disordered phenotype. So we'll talk about um, why that is in a moment. Now we see here that when a child is connected, the shape is directly connected to a new shape. That means that the shape, um, uh, it means that the child married someone from outside the family. And we see here that they had children, and the grandchildren, they had two children. Neither granddaughter has the disordered phenotype. But we see here that when the granddaughter married someone or had or hold on, gotta pause this. Sorry about that. It's Valentine's Day, and I got a call from my mom. All right, cool. So, all right, let's go back and let's review this once more because I have a feeling I said it in a very confusing manner. All right, well, actually, why show you pictures? Let's go ahead and draw it out. Because what I would like you to do is be able to identify between two types of pedigree charts: autosomal dominant and autosomal recessive. The PowerPoint talks about X-linked, just ignore that. So once again, a pedigree chart shows the prevalence of one disorder being in a family lineage. So let's go ahead and draw a pedigree chart here. Circle refers to females, squares refer to males. Whenever a shape is blended in, it means that they have the disordered phenotype. So they're showing the phenotype. Now, when a line goes down, it connects to other shapes. So this refers to the offspring. They have three offspring. Neither offspring is showing the disordered phenotype. And we're going to say that this one's going to marry in, have offspring. Now, if you see a pedigree chart that looks like this is indicative of what we call an autosomal recessive pedigree chart. This means that the disorder that runs in this family, whatever it is recessive in nature. Now, how do we know? For one, we look for, does the disorder skip generations? What this means is that the disorder is present in one generation, but not the next. So if we're going to label the generations, so this here is generation 1, this here is generation 2, this here is generation 3. So we see the disorder is present in generation 1, excuse me, but not generation 2, but we see it again in generation 3. In case you're curious why, if we are correct and this is autosomal recessive, then that means that this parent must have this genotype and this parent must have this phenotype. And if that's the case, they're going to have 100% carriers. Now you might be asking, well, how do we know that this parent is in a heterozygote? The answer is we don't, but this is an introductory class, so I keep things on the simpler side. Okay, now since the disorder is present in the offspring, then we have to also assume that this individual that married in has to also be a carrier, because that's the only way that two individuals that do not have the phenotype in question can have an offspring that has the disorder. So another sign that this is an autosomal recessive disorder is the question: do two phenotypically normal parents have an offspring with the condition? Okay, if the answer is yes, then it is recessive. And also, do less than half of the family members have the condition? If the answer is yes, then it is going to be autosomal recessive. Now, how would it look if it was autosomal dominant? Well, let's go find out. Now, if it is going to be autosomal dominant, it's going to instead look more like this. So, autosomal dominant. Now, in this case, with autosomal dominant, unlike recessive, it does not. So the question is, does the disorder skip generation? Does not skip generations. So you see it present in every generation. Do two phenotypically normal parents have an offspring with the condition? No, that cannot happen. Only parents. Actually, let me make this simpler. One parent must have the recessive phenotype. Not recessive. What am I doing? Must have the affected phenotype. So one parent must have the affected phenotype. And also, usually more than half display the recessive, not recessive, what am I doing? The affected phenotype. I'm going through this quickly because subconsciously I know that we've been at this now for like an hour and a half, and I'm sure you're probably more tired of hearing my voice than I am. Actually, you know what? I like to hear my voice. I have a very nice, I've been told I have a very calming presence. So I need to do a better job of my calming presence being calming for me. But that probably would be better if I did not just down one of these Celsius heats. So I did not plan this joke, but when I first started teaching at the high school of math, is they did an assembly where they were supposed to imitate a teacher, and the audience would have to find out or guess who the teacher was. Now, keep in mind, at the time I was new, a new teacher, and the students, I didn't really know if they picked up on my mannerisms yet. And all of a sudden, a student, she shows up on stage with a can of Monster Energy, and instantly, students are like, "Mr. Thomas, Mr. Thomas." I'm like, "All right, well, I guess they picked up on that habit." Yeah, when I was teaching at a school called St. Genevieve, and I didn't think much of this. I should have. Is I had a protein shake that was I would bring protein shakes to school because we weren't allowed to eat in the buildings, and I would freeze them, and they would get um, pretty cold. So I would just have it in a paper bag with a straw, and I'm thinking nothing of it. Just have my protein shake in a paper bag. Did the job. And one day a student goes, "Mr. Thomas, what's in the bag?" And I think they thought I was taking swigs of vodka or something in between my lectures. So yeah, I don't do the paper bag anymore. All right, yeah, so more than half display the affected phenotype, or at least half. We see here that in this family, we see that 4 of them are expressing um, the affected phenotype, and half of them are not. All right, so what I'll expect you on your quiz or exam is to be able to identify the difference between autosomal dominant and autosomal recessive pedigree charts, as we call this. You also have to identify the different types of inheritance and be able to do simple monohybrid problems using either dominant and recessive inheritance, incomplete dominance, or codominance. Also, it's important that you understand the terminology. You also need to identify examples of pletrophy and polygenic inheritance. All right, so if there's any questions, feel free to reach out. If not, it's been an honor teaching you classic genetics, one of my favorite topics in all of biology. And I'll see you again for next lecture, molecular genetics.